Every living thing you have ever seen, touched, or heard of depends on water. Every cell, every organism, every ecosystem on this planet is built around one molecule: H2O. But here is the question nobody asked for billions of years. What if water is not special? What if life simply uses water because it happens to be everywhere on Earth, not because it is the only option? Scientists are now exploring something that challenges everything we thought we knew about biology. Liquid solvents that exist at temperatures where water turns to ice. Chemical environments so hostile they would dissolve your body in seconds. Worlds where the oceans are not water, but methane, or ammonia, or even sulfuric acid. And in those alien seas, something might be moving, growing, evolving. Something we would not recognize as life, but life nonetheless. This is not science fiction. These are real places in our solar system, right now. And the chemistry that could support life there is stranger than anything you have imagined. To understand why water seems irreplaceable, you need to see what it actually does inside a living cell. Water is not just a background liquid that fills space. It is an active participant in nearly every biological process that keeps you alive. At the molecular level, water has a peculiar structure. The oxygen atom pulls electrons toward itself, creating a slight negative charge on one end and a slight positive charge on the other. This polarity turns each water molecule into a tiny magnet, capable of grabbing onto other charged molecules and pulling them apart. When you eat food, enzymes break down complex molecules through a process called hydrolysis. A water molecule literally splits the chemical bonds holding proteins, fats, and carbohydrates together. Without water, your body cannot extract energy from food. It just sits there, chemically inert. Water also stabilizes proteins. These molecules fold into precise three-dimensional shapes, and water molecules surround them, holding them in place through hydrogen bonds. Change the temperature too much, and those bonds break. The protein unfolds. It stops working. This is why a fever can be dangerous and why extreme heat kills. But here is what makes water truly exceptional. It has an absurdly high specific heat capacity. This means water can absorb massive amounts of thermal energy without its temperature changing much. Your body is roughly 60% water by mass, and that water acts like a thermal buffer. When the environment heats up, your internal chemistry does not immediately spiral out of control. Water absorbs the excess energy, keeping your enzymes and cellular machinery operating within safe limits. On a planetary scale, oceans do the same thing. They regulate Earth's climate, preventing the wild temperature swings that would make complex life nearly impossible. Water also has another rare property. It is less dense as a solid than as a liquid. Ice floats. This seems trivial until you realize what it means for life. In winter, lakes freeze from the top down, creating an insulating layer that protects aquatic organisms below. If ice sank, every body of water would freeze solid from the bottom up. Life as we know it would not exist in temperate or cold regions. The chemistry of water is so finely tuned to support biology that for centuries, scientists assumed it was the only option. But that assumption is cracking. The first challenge to water's monopoly came from a simple observation. Life on Earth survives in places it should not. Deep beneath the ice in Antarctica, microbes live in lakes that have been sealed off from the surface for millions of years. Near hydrothermal vents on the ocean floor, organisms thrive in water superheated to hundreds of degrees Celsius. In the Atacama Desert, some of the driest places on the planet, bacteria endure with almost no water at all. These extremophiles do not just tolerate harsh conditions. They thrive in them. And their existence forces a question. If life can adapt to environments this extreme on Earth, why couldn't it adapt to completely different solvents on other worlds? This is where astrobiologists step in. They are not asking whether life needs water. They are asking what defines a solvent capable of supporting life. The answer comes down to a few key criteria. First, the liquid must remain stable over a reasonable temperature range. If it boils away or freezes solid too easily, complex chemistry cannot happen. Second, it must be a good solvent. It needs to dissolve and transport the molecules that life uses for metabolism, structure, and replication. Third, it must be abundant. A rare liquid that only exists in tiny pockets is not going to create thriving ecosystems. And fourth, it must allow for controlled chemical reactions. Life is not random. It is organized chemistry, and the solvent needs to enable that organization without destroying the molecules involved. Water meets all these criteria perfectly. But it is not the only substance that does. Ammonia has been on the list of alternative solvents for decades. Chemically, ammonia is similar to water. It is polar, which means it can dissolve ionic compounds and form hydrogen bonds, though those bonds are weaker than the ones in water. Liquid ammonia exists at much lower temperatures than water. It remains fluid between -78 degrees Celsius and -33 degrees Celsius. This opens up an entirely new range of habitable environments. Planets and moons far from their stars, places where water would be frozen solid, could have liquid ammonia oceans. Ammonia is also incredibly common in the universe. It is the fourth most abundant molecule in space. Anywhere you find nitrogen and hydrogen, which are among the most common elements, you can form ammonia. This means that ammonia-based life would not be limited to rare, exotic worlds. It could exist on a significant fraction of cold planets and moons across the galaxy. But ammonia-based life would not look like anything on Earth. The lower temperatures would slow chemical reactions dramatically. Metabolism would be slower. Growth would be slower. Evolution itself would operate on different timescales. The molecules that make up ammonia-based organisms would also be different. Proteins as we know them might not work. Lipids that form cell membranes in water might not function in ammonia. Life would have to invent entirely new molecular architectures. This is not a problem. It is an opportunity. Evolution does not care about our expectations. It optimizes for survival in whatever environment exists. If ammonia is available and stable, life will find a way to use it. Now shift your attention to Saturn's moon Titan. This is the only place in the solar system, other than Earth, where liquids pool on the surface. But those liquids are not water. Titan's surface temperature hovers around -179 degrees Celsius. At that temperature, water is as hard as granite. What flows on Titan is methane and ethane. Lakes, rivers, and seas of liquid hydrocarbons stretch across its surface, fed by rain from its thick, nitrogen-rich atmosphere. For years, scientists dismissed Titan as too cold for life. Then they started running the numbers. Methane and ethane are nonpolar solvents. They do not dissolve the same compounds that water does, which means life in Titan seas would need completely different chemistry. In 2015, researchers proposed a theoretical cell membrane called an azotosome. Unlike Earth cells, which use phospholipid bilayers, azotosomes would be made from acrylonitrile, a compound detected in Titan's atmosphere. Computer simulations suggested these structures could remain stable in liquid methane, forming flexible, self-sealing membranes similar to biological cells. The discovery was exciting, but follow-up experiments revealed complications. When researchers tested mixtures of liquid ethane and vinyl cyanide, the molecules formed crystals instead of bubbles. No azotosomes appeared. This does not mean life cannot exist on Titan. It means we do not yet understand how it would work. The chemistry is more complex than early models suggested, and the energy sources are still unclear. One candidate is acetylene, a hydrocarbon that could serve as food for methane-based organisms. Another is hydrogen. Titan's atmosphere produces hydrogen through photochemical reactions, and that hydrogen could be consumed by life in a process similar to how we use oxygen. Data from a Cassini mission showed something strange. Hydrogen near Titan's surface is disappearing. Acetylene levels are also lower than expected. One interpretation is that something is consuming them. Not geological processes. Not atmospheric chemistry. Something biological. This is not proof. It is a hint. A possibility. And that possibility is enough to keep scientists looking. What makes Titan so compelling is not just the presence of liquid. It is the chemistry happening in that liquid. Complex organic molecules are forming. Reactions are occurring that we do not fully understand. And in that chemical complexity, there is room for life. Not life as we know it, but life adapted to an environment where methane rains from the sky and oceans freeze at temperatures that would shatter steel. If you thought methane seas were strange, consider Venus. The surface is a hellscape. Temperatures exceed 450 degrees Celsius. The atmospheric pressure is 90 times that of Earth. Lead would melt into puddles. But 50 kilometers above the surface, in the cloud layer, conditions are surprisingly mild. Temperatures range from 0 to 60 degrees Celsius. Pressure is roughly equivalent to sea level on Earth. And the clouds are made of concentrated sulfuric acid. Sulfuric acid should destroy organic molecules. It is one of the most corrosive substances known. Yet recent experiments tell a different story. Researchers placed amino acids, the building blocks of proteins, into sulfuric acid solutions matching the concentration found in Venus's clouds. After 4 weeks, 19 out of 20 amino acids remained stable. Their molecular backbones did not break down. They did not dissolve into unrecognizable fragments. They survived. This does not mean life exists in Venus's clouds, but it means life could exist there. Sulfuric acid, despite its reputation, might support a specialized biochemistry. Organisms would need acid-resistant membranes, enzymes that function in extreme acidity, and metabolic pathways completely unlike anything on Earth. But those are engineering problems, not fundamental barriers. The possibility is real. Venus's clouds also contain another intriguing element: phosphine. In 2020, scientists detected phosphine gas in Venus's atmosphere. On Earth, phosphine is almost exclusively produced by biological processes or industrial activity. There are no known geological processes on Venus that should generate phosphine in the quantities detected. The discovery sparked intense debate. Some argued the signal was misinterpreted. Others claimed the detection was real but the source was unknown chemistry, not biology. The question remains unresolved. But what matters is this: Venus, a world we once dismissed as completely sterile, now sits on the list of places where life might exist. Not on the surface. Not in liquid water. but floating in sulfuric acid clouds, sustained by chemistry we barely understand. These examples share a common thread. Life, if it exists beyond Earth, will not follow our rules. It will follow the rules of its environment. And those environments are more varied than we ever imagined. But what about the building blocks themselves? Water is not just a solvent. It is a participant in Earth's biochemistry. It breaks apart molecules through hydrolysis. It donates hydrogen atoms. It stabilizes protein structures. If life uses a different solvent, does it also need different chemistry? The answer is almost certainly yes. Consider silicon. On the periodic table, silicon sits directly below carbon. Both elements have 4 valence electrons, allowing them to form 4 chemical bonds. Carbon forms the backbone of every organic molecule on Earth. Could silicon do the same? The idea has fascinated scientists for decades. Silicon is abundant. Earth's crust contains 925 times more silicon than carbon. If life could use silicon instead of carbon, the universe would be full of potential building blocks. But there is a problem. When carbon reacts with oxygen, it forms carbon dioxide, a gas that dissolves in water and can be exhaled or released. When silicon reacts with oxygen, it forms silicon dioxide. Silica. A solid. This difference is fundamental. Carbon-based life can easily process and eliminate waste. Silicon-based life would constantly produce solid byproducts that clog its internal systems. In a water-rich environment, silicon chemistry collapses almost immediately. Silicon dioxide is stable, insoluble, and inert. It does not participate in the kind of dynamic, reversible reactions that life requires. But remove water from the equation, and silicon becomes interesting again. In environments dominated by sulfuric acid or other non-aqueous solvents, silicon compounds behave differently. Organosilicon molecules, compounds where silicon bonds with carbon and hydrogen, remain stable. They do not immediately oxidize into inert solids. In such an environment, silicon could theoretically form the backbone of complex molecules. Not in the same way carbon does, but in a way that works under those specific conditions. This is speculative. No one has observed silicon-based life. But the chemistry is plausible. And that is enough to keep the idea alive. Then there is the question of energy. Life needs energy to maintain itself, grow, and reproduce. On Earth, most life gets energy from chemical reactions involving oxygen. Plants use sunlight to convert carbon dioxide and water into glucose. Animals eat plants or other animals and extract energy through respiration. But oxygen is not the only option. Some of Earth's most ancient organisms, bacteria living near hydrothermal vents, extract energy from hydrogen sulfide or methane. They do not breathe oxygen. They never see sunlight. They exist in complete darkness, sustained by chemistry alone. These organisms prove that life can function without the energy sources we consider essential. And if life on Earth can survive without oxygen or sunlight, life elsewhere might use entirely different energy pathways. On Titan, acetylene or hydrogen could serve as fuel. On Venus, ultraviolet light filtering through sulfuric acid clouds might drive photochemical reactions. On Europa, Jupiter's icy moon, life might extract energy from chemical gradients created by hydrothermal vents beneath kilometers of ice. Europa deserves its own attention. The surface is frozen solid. temperatures reach -160 degrees Celsius. But beneath that ice lies an ocean. A global ocean, potentially containing more liquid water than all of Earth's oceans combined. The ice shell is estimated to be 15 to 25 kilometers thick. Below that, liquid water. And at the bottom of that ocean, there may be hydrothermal vents, similar to those found on Earth's ocean floors. These vents release heat and chemicals from the Moon's rocky interior. On Earth, hydrothermal vents support entire ecosystems. Tube worms, shrimp, bacteria. Life thrives in total darkness, sustained by chemical energy alone. If Europa has similar vents, it could support similar life. Not identical. Europa's ocean is likely more acidic or alkaline than Earth's. The pressure is different. The chemistry is different. But the principle is the same. Chemical energy flowing from the interior of a celestial body into a liquid medium. That is all life needs. Enceladus, another moon of Saturn, offers an even more tantalizing case. Enceladus is small. Only 500 kilometers in diameter. But it has a subsurface ocean. We know this because Enceladus is geologically active. Massive geysers erupt from its south pole, shooting water vapor, ice particles, and organic molecules into space. The Cassini spacecraft flew through these plumes and analyzed their composition. The results were striking. The plumes contain molecular hydrogen. Hydrogen is a sign of hydrothermal activity. It forms when hot water reacts with rock in a process called serpentinization. This process releases hydrogen gas, which can be used by certain microbes as an energy source. On Earth, methane-producing bacteria, called methanogens, consume hydrogen and carbon dioxide to produce methane and water. This reaction releases energy, which the bacteria use to survive. The same process could occur on Enceladus. The plumes also contain complex organic molecules. Not just simple compounds like methane or ammonia. Complex carbon-based molecules. Nitrogen-bearing compounds. Oxygen-bearing compounds. Molecules that, on Earth, are associated with biological processes. This does not prove life exists on Enceladus. But it proves the ingredients are there. Liquid water. Chemical energy. Organic molecules. Everything life needs. What makes Enceladus particularly interesting is accessibility. The plumes shoot material directly into space. We do not need to drill through kilometers of ice. We do not need to land on the surface. We can fly a spacecraft through the plumes, collect samples, and analyze them. Future missions could carry instruments sensitive enough to detect biosignatures. Amino acids. Lipids. Even fragments of genetic material. If life exists in Enceladus's ocean, the plumes might deliver the evidence directly to us. But detecting life in these environments is not straightforward. On Earth, we recognize life because we know what to look for. Cells. DNA. Proteins. Metabolic byproducts. But life using a different solvent, different chemistry, different energy sources might not produce the same biosignatures. It might not have DNA. It might not have cells with lipid membranes. It might not leave behind the same chemical traces. This creates a fundamental problem. How do you search for something when you do not know what it looks like? The answer is to focus on universal principles. Life, regardless of its chemistry, must have certain characteristics. It must extract energy from its environment. It must maintain an organized internal structure separate from its surroundings. It must reproduce or replicate in some form. And it must evolve, adapting to changing conditions over time. These are not specific to carbon or water. They are requirements for any system we would call alive. If we find something in Titan's seas or Venus's clouds that consumes energy, maintains structure, and reproduces, we would recognize it as life. Even if its chemistry is completely alien. The search for non-water life forces us to rethink what life is. For centuries, we defined life based on what we observed on Earth. Carbon, water, oxygen, DNA. But those are not definitions. They are examples. Life is not a specific molecule or a specific process. Life is a phenomenon. A pattern of organization that persists, adapts, and evolves. That pattern can emerge in many forms. And the more we explore, the more forms we discover are possible. Venus presents a different extreme. The surface is a hellscape. Temperatures reach 900 degrees Fahrenheit. Atmospheric pressure is 90 times that of Earth. Lead would melt into pools on the ground. But 50 kilometers above that inferno, conditions change. The clouds of Venus exist in a narrow band where temperatures hover around 80 degrees Fahrenheit. The pressure is similar to Earth's surface. You could stand there, theoretically, without a pressure suit. The problem is the chemistry. Venus's clouds are made of concentrated sulfuric acid. Droplets of acid so corrosive they would dissolve most materials in seconds. On Earth, sulfuric acid is a tool for breaking things down. It destroys organic molecules. It strips apart chemical bonds. The idea that life could exist in such an environment seems absurd. But recent research suggests otherwise. Scientists tested amino acids, the building blocks of proteins, in sulfuric acid concentrations matching Venus's clouds. After 4 weeks, 19 of 20 amino acids remained stable. Their molecular backbones intact. Some underwent modifications, but they did not disintegrate. This does not mean life exists in Venus's clouds. But it means the door is not closed. If amino acids can survive, perhaps other organic molecules can too. Perhaps a completely different biochemistry could emerge. One where sulfuric acid is not a threat but a medium. Sulfuric acid is a powerful solvent. It dissolves many substances water cannot touch. It remains liquid across a wide temperature range, from -30 degrees Celsius to over 300 degrees Celsius. And unlike water, it is a protonating solvent. It donates protons aggressively, creating an environment where certain chemical reactions occur more readily than they would in water. Life adapted to this environment would not resemble Earth life. Its membranes would need to resist acid. Its proteins would require different structures. Its metabolism would extract energy from chemical reactions that, to us, seem destructive. But the chemistry is theoretically possible. And Venus's clouds offer another advantage. They are dynamic. Updrafts and downdrafts cycle material through different temperature and pressure zones. This creates opportunities for complex chemistry. Prebiotic molecules could form in cooler regions, then be transported to warmer zones where reactions accelerate. Over time, this cycling could drive chemical evolution. Not in the same way it happened on Earth, but through a parallel process shaped by Venus's unique conditions. The idea gains weight when we consider extremophiles on Earth. Organisms that thrive in environments we once thought uninhabitable. Bacteria living in hot springs with pH levels below 1. Archaea flourishing in boiling acidic lakes. These creatures do not just tolerate extreme conditions. They require them. Their enzymes only function in high acidity. Their membranes only remain stable at extreme temperatures. Remove them from their environment, and they die. If life on Earth can adapt to acid, why not life elsewhere? The real question is whether chemistry alone can give rise to biology in such an environment. Or whether water is not just useful but essential for the origin of life itself. Mars offers yet another scenario. The surface is cold and dry. Temperatures average -80 degrees Fahrenheit. The atmosphere is thin, mostly carbon dioxide. Liquid water cannot exist on the surface for more than a few seconds before it freezes or evaporates. But Mars was not always like this. Billions of years ago, it had rivers, lakes, possibly oceans. A thicker atmosphere. A warmer climate. If life emerged during that time, it may have found ways to survive as the planet changed. One hypothesis involves hydrogen peroxide. A mixture of water and hydrogen peroxide, about 60% peroxide by mass, has a freezing point of -70 degrees Celsius. It does not crystallize easily. It supercools, remaining liquid even when temperatures drop below freezing. And it is hygroscopic. It attracts water molecules from the atmosphere. On a planet where water is scarce, this property is invaluable. Organisms using a water peroxide mixture as their internal fluid could survive in Martian soil. They could extract water vapor from the air. They could remain active at temperatures that would kill most Earth life. Hydrogen peroxide also provides oxygen. Breaking it down releases molecular oxygen, which could be used in metabolism. This would solve two problems at once. A solvent that stays liquid in the cold and a source of oxidizing power. Does this mean life on Mars uses hydrogen peroxide? We do not know. But when NASA's Viking landers tested Martian soil in the 1970s, they found strange results. Soil samples released oxygen when exposed to nutrients. Some scientists interpreted this as a sign of biological activity. Others argued it was just chemistry. Peroxides in the soil reacting with the test compounds. The debate remains unresolved. Future missions might provide answers. But the Viking results remind us that chemistry on other planets does not always behave as we expect. These scenarios, Venus, Mars, Titan, and Enceladus, are not just thought experiments. They represent real places in our solar system where the conditions for alternative biochemistry exist. But identifying life in these environments requires new strategies. On Earth, we look for familiar markers. DNA sequences. Cell structures. Specific metabolic byproducts. But non-water life might not produce any of these. It might not have DNA. It might not have cells as we define them. Its waste products might be molecules we have never encountered. This creates a detection problem. How do you recognize life when it does not look like life? One approach focuses on disequilibrium. Life creates chemical imbalances. It consumes certain molecules and produces others. It maintains an internal environment different from its surroundings. On Earth, oxygen in the atmosphere is a biosignature. Oxygen is reactive. It should not persist in large quantities unless something continuously produces it. That something is photosynthesis. On another world, we might look for similar imbalances. An excess of hydrogen on Titan. Unexpected organic molecules in Venus's clouds. Chemical gradients in Enceladus's plumes. These would not prove life exists. But they would suggest something unusual is happening. Something worth investigating further. Another approach examines complexity. Life produces complex molecules. Not just large molecules, but molecules with specific patterns. Information-carrying molecules like DNA. Catalytic molecules like enzymes. Structural molecules like proteins. Abiotic chemistry can create complex molecules too. But they tend to be random. A mix of many different structures without order. Biology produces order. It makes the same molecules repeatedly. It builds structures with precise arrangements. If we found this kind of molecular order in a sample from Titan or Enceladus, it would be a strong hint. Not proof, but a reason to look deeper. The challenge is that our instruments are designed to detect Earth-based biosignatures. They search for amino acids in specific configurations. They look for lipids with particular structures. They are optimized for water-based biochemistry. Detecting life that uses different chemistry requires different tools. Instruments that can identify patterns rather than specific molecules. Sensors that measure energy flows and chemical gradients. Techniques that recognize organization and complexity without assuming what form that organization will take. We are starting to develop these tools. But we are still in the early stages. The philosophical implications are profound. For all of human history, we have had one example of life. Birth. One chemistry. Carbon and water. One genetic system. DNA and RNA. This single example has shaped our entire understanding of biology. We define life based on what we observe here. But what if Earth life is not the template? What if it is just one variation among countless possibilities? The discovery of non-water life would force a complete reconceptualization. It would prove that life is not a specific set of molecules but a general phenomenon. A pattern that emerges when energy flows through matter under the right conditions. The molecules do not matter. The solvent does not matter. What matters is the organization. The ability to extract energy, maintain structure, reproduce, and evolve. If we found life in Titan's methane lakes, it would answer one of science's oldest questions. Are we alone? But it would also raise new ones. How common is life in the universe? How many forms can it take? Is intelligence inevitable, or is it a rare accident? And perhaps most unsettling, how many forms of life could exist that we would not even recognize? Life so different from ours that we might walk past it, sample it, analyze it, and still not realize what we were looking at. The real terror is not that we might miss alien life. It's that we might destroy it before we know it's there. Every mission we send carries contamination. Earth microbes hitching a ride on metal surfaces. Organic molecules from fuel and lubricants. Chemical residues from manufacturing. We sterilize our spacecraft, but sterilization is never perfect. A few hardy organisms always survive. Extremophiles that can endure radiation, vacuum, temperature swings. They travel with us. And if they reach an environment where they can function, even barely, they might grow. They might spread. They might outcompete whatever fragile chemistry already exists there. This is called forward contamination. It's one of planetary science's greatest concerns. We have protocols. Strict guidelines about where we can land and what we can touch. But protocols are only as good as our understanding. And our understanding is built on Earth life. We know how to kill Earth bacteria. We know what temperatures and chemicals will destroy our kind of biology. But what about theirs? Imagine a probe landing on Titan. It carries microbes in microscopic cracks. Those microbes are water-based. They cannot survive in liquid methane. They die almost instantly. No problem. But what if the probe also carries trace amounts of water? Just a few molecules trapped in a seal or coating. That water melts. It mixes with methane. It creates a microenvironment. And in that tiny pocket, something unexpected happens. Earth chemistry and Titan chemistry interact. They interfere with each other. They create compounds that would not form naturally on either world. We just contaminated an alien ecosystem. Not with life. With chemistry. And we might never know we did it. The reverse problem is equally serious. Backward contamination. Bringing alien material back to Earth. This sounds like science fiction. But it's a real concern for sample return missions. Mars samples. Europa ice. Enceladus plume particles. If any of these contain life, even life completely unlike ours, we need to be careful. Not because it might be dangerous, but because it might be vulnerable. Or because it might interact with Earth's biosphere in ways we cannot predict. The Apollo missions brought back moon rocks. The astronauts were quarantined. The samples were isolated. This was probably unnecessary. The moon is sterile, but it established a precedent. When we bring back samples from worlds that might harbor life, the protocols will be far stricter. The samples will be studied in sealed facilities. They will never touch Earth's atmosphere. They will be analyzed remotely, through robotic systems. And if we find something living, we will face an ethical dilemma. Do we study it? Do we try to keep it alive? Do we destroy it to prevent any possibility of contamination? These questions have no easy answers. They require us to think carefully about what we value. Scientific knowledge. The preservation of alien ecosystems. The protection of Earth's biosphere. Sometimes these values conflict. But there's another layer to this. One that's even more unsettling. What if life is common, but intelligence is not? What if the universe is full of microbial ecosystems, chemical networks, self-organizing systems that technically qualify as alive, but never develop awareness? Never build technology. Never ask questions. We would be alone in a different way. Surrounded by life, but still the only minds. The only consciousness. The only beings capable of understanding what life even is. That's a heavy responsibility. It means that if we find microbial life on another world, we are not just discovering it. We are also its only witness. Its only interpreter. Perhaps its only protector. Or maybe the opposite is true. Maybe intelligence is inevitable. Maybe any system that can evolve, given enough time, eventually produces minds. This would mean we are not special. Just early. The universe is young. Most stars have not yet formed. Most planets have not yet cooled. We might be among the first intelligent species to emerge. And if we are, that gives us another responsibility. To preserve the conditions that allow life to continue. To avoid contaminating worlds where evolution is still unfolding. To leave space open for whatever comes next. The search for extraterrestrial intelligence has been running for decades. We have listened for radio signals. We have searched for laser pulses. We have analyzed data from millions of star systems. And we have found nothing. No deliberate transmissions. No megastructures. No signs of technology. This is called the Fermi paradox. If intelligent life is common, where is everybody? One possible answer is that we are looking for the wrong thing. We are searching for technological signatures. Radio waves. Heat signatures. Atmospheric pollution. But what if intelligence does not always lead to technology? What if some minds develop without tools? Without language? Without the drive to manipulate their environment? They might be intelligent in ways we would not recognize. Self-aware. But silent. What if consciousness is common, but communication is rare? Another answer is that technological civilizations destroy themselves. They develop too quickly. They exhaust their resources. They poison their planet. They go extinct before they can spread beyond their home world. If this is true, then the silence of the universe is a warning. A message written in absence. It tells us that survival is not guaranteed. That intelligence alone is not enough. That we need wisdom as well. Or maybe we are simply early. The universe is 13.8 billion years old. But the first generation of stars contained no heavy elements. No carbon. No oxygen. No silicon. Planets like Earth could not form. Life as we know it could not exist. It took billions of years for enough stars to die and release their elements into space. For new stars to form from enriched gas. For rocky planets to coalesce. Earth is 4.5 billion years old. Life appeared almost immediately. But complex life took much longer. Multicellular organisms did not emerge until 600 million years ago. Intelligence is even more recent. Modern humans have existed for only 300,000 years. Technology for 10,000. Radio for 100. If this timeline is typical, then we might be among the very first technological species. The universe might be waiting for others to catch up. Still, there's one more possibility. One that challenges everything. Maybe life is common. Maybe intelligence is common. But maybe the kind of intelligence that builds radio telescopes and sends messages into space is vanishingly rare. Maybe most intelligent species are content. They do not need to explore. They do not need to expand. They live in balance with their environment. they never develop the restless curiosity that drives us to ask these questions. They exist. They think. But they do not reach out. And so we never find them. If that's true, then our drive to explore makes us unusual. Not superior. Just different. And that difference might be the reason we are the ones searching. The ones wondering. The ones building spacecraft and pointing telescopes at distant stars. We are not looking for life like us. We are looking for any sign that we are not alone in caring about the answer. Back to the practical question. How do we search for life we might not recognize? We start by expanding our definition. Stop looking for specific molecules. Start looking for patterns. Complexity. Organization. Energy flow. Chemical disequilibrium. These are the universal signatures. They do not depend on water or carbon. They apply to any system that maintains itself against entropy. Anything that consumes energy to build structure. Anything that reproduces information. This requires new instruments. Sensors that measure gradients rather than concentrations. Spectrometers that identify patterns rather than peaks. Microscopes that recognize organization at any scale. We are building these tools. But slowly. Because most funding still goes to missions designed to find Earth-like life. Water. Organic molecules. Amino acids. These missions are important. They will answer crucial questions. But they will not tell us if life can exist in radically different forms. The next generation of missions will be different. They will target worlds we currently consider marginal. Titan. Enceladus. Venus's clouds. They will carry instruments designed to be agnostic. To measure without assuming. To detect the unexpected. And if they find something, it will not be a triumphant moment. Not immediately. It will be a moment of confusion. Of doubt. Of careful analysis. Because recognizing truly alien life will be harder than finding it. We will need to prove that what we are seeing is not geology. Not chemistry. But something more. Something that fits our expanded definition of living. Self-sustaining. Evolving. Organized. And when we finally make that determination, everything changes. Not just for humanity. For the entire concept of life itself. Because if we confirm that biology can arise independently, that it can take forms we never predicted, that it operates on principles we never imagined, then we have proven something profound. Life is not an accident. It is an inevitable consequence of chemistry and energy. It emerges wherever conditions allow. And the universe becomes a fundamentally different place. One filled with possibilities we are only beginning to comprehend. Consider what this means practically. Right now, we classify planets as habitable based on narrow criteria. Liquid water. Moderate temperature. Rocky surface. Protective atmosphere. This framework eliminates most worlds immediately. Gas giants. Ice worlds. Airless rocks. Planets orbiting too close or too far from their stars. But if life can use ammonia instead of water, suddenly frozen moons become candidates. If it can metabolize hydrogen instead of oxygen, gas giant atmospheres become ecosystems. If it can build cells from silicon compounds in sulfuric acid, Venus transforms from hell to habitat. The number of potentially living worlds explodes. Not by a factor of 2 or 3, by orders of magnitude. We already have targets that challenge our assumptions. Titan stands out immediately. Saturn's largest moon has lakes and rivers. Not of water. Of liquid methane and ethane. The temperature hovers around -290 Fahrenheit. Water ice behaves like bedrock. Any life here would need completely different chemistry. In 2015, researchers proposed azotosomes, theoretical cell membranes made from vinyl cyanide. These structures could function in liquid methane the way phospholipid bilayers work in water. Early observations suggested vinyl cyanide existed in Titan's atmosphere, enough to form billions of cell-like structures in every cubic centimeter of liquid. But later experiments threw cold water on the idea. Or rather, cold methane. When scientists mixed vinyl cyanide with liquid ethane in the lab, they did not get bubbles. They got crystals. The molecules locked together in solid lattices instead of flexible membranes. No azotosomes formed in pure methane either. Does this kill the possibility of methane-based life? Not even remotely. It eliminates one specific hypothesis. But Titan's chemistry is staggeringly complex. The Cassini mission detected dozens of organic molecules in the atmosphere. Benzene, propane, hydrogen cyanide, complex polymers called tholins that rain down onto the surface. These compounds accumulate in the lakes. They react. They concentrate. Something is happening there. Whether it qualifies as life remains unknown. But dismissing Titan because one membrane model failed would be like abandoning Earth because the first origin of life experiment did not produce DNA. Astrobiologist Chris McCabe proposed a testable prediction back in 2005. If methane-based life exists on Titan, it should consume hydrogen and acetylene. Both are produced abundantly in the upper atmosphere by sunlight breaking apart methane. Both should accumulate at the surface. But measurements show acetylene levels far lower than expected. Hydrogen too. One interpretation, something is eating them. Using acetylene for energy the way we use glucose. Breathing hydrogen the way we breathe oxygen. The chemistry works. The thermodynamics check out. The only question is whether biology is actually doing it. We need to go back. The Dragonfly mission, scheduled to launch in 2027, will do exactly that. A nuclear-powered quadcopter will fly across Titan's surface, landing in different locations, sampling the chemistry, looking for patterns that suggest metabolism, not searching for specific molecules, searching for disequilibrium, for ratios that should not exist unless something is actively maintaining them. If we find those patterns, we face the recognition problem all over again, proving that chemistry alone cannot explain what we are seeing, demonstrating that organization has crossed the threshold into biology. Venus presents an even stranger scenario. The surface is an oven. 900 degrees Fahrenheit. Atmospheric pressure 90 times Earth's. Lead melts there. But 50 kilometers up, in the thick cloud layer, conditions moderate dramatically. Temperature drops to room levels. Pressure matches sea level on Earth. And the clouds themselves are made of concentrated sulfuric acid. Droplets so corrosive they would dissolve human tissue in seconds. Yet in 2020, researchers found something unexpected. 19 amino acids remained stable in concentrated sulfuric acid for at least 4 weeks. Their molecular backbones did not break down. 8 of them changed slightly. 11 showed no reaction at all. This does not prove life exists in Venus's clouds. But it demolishes the assumption that sulfuric acid automatically destroys organic chemistry. The clouds also contain anomalies. Phosphine was detected in 2020. A gas produced on Earth almost exclusively by biological activity. The detection sparked immediate controversy. Other teams questioned the data, suggested alternative explanations, proposed abiotic chemical pathways. The debate continues. But even if phosphine is not there, something else is. Dark patches in the clouds that absorb ultraviolet light. They appear and disappear. They move with atmospheric circulation. Their composition remains unknown. Iron compounds could explain them. Sulfur allotropes. Or something metabolizing sulfur dioxide and producing elemental sulfur as waste. We do not know. And we will not know until we send a mission capable of sampling the clouds directly. Mars offers a different puzzle. The Viking landers in 1976 conducted experiments designed to detect microbial life. One test showed positive results. Martian soil released radioactive carbon dioxide when exposed to nutrients. Exactly what would happen if microbes were metabolizing the food. But another test found no organic molecules. The conclusion: chemistry, not biology. Oxidizing compounds in the soil mimicked biological reactions. Case closed. Except the case may not be closed. In 2007, researchers proposed that Martian organisms might use a water-hydrogen peroxide mixture as their internal fluid. This mixture stays liquid down to -60 degrees Celsius. It attracts water from the atmosphere. And it provides oxygen directly to cells. Organisms using this chemistry would not look like Earth life. They might even be destroyed by the Viking experiments, which used liquid water. Adding water to a hydrogen peroxide-based cell would be like drowning a human in acid. The proposal remains speculative. But it highlights how our methods can bias results. If we search only for water-based life, we will only find water-based life. If it exists at all, everything else becomes invisible. Not because it is not there. Because we are not looking in the right way. Earth itself provides clues about how far life can bend the rules. Tardigrades, microscopic animals less than a millimeter long, survive complete dehydration. They enter a state called ton. Their bodies collapse. Metabolism stops entirely. They can remain this way for decades. When water returns, they rehydrate and resume normal activity within hours. Recent research identified the proteins that make this possible. Tardigrade disordered proteins. These molecules have no fixed structure. They shift shape based on their environment. When water disappears, they form protective networks around cellular machinery. Glass-like structures that preserve everything in suspended animation. When water returns, they dissolve. The cell restarts. This is not just surviving without water. This is thriving by abandoning water entirely during stress, then reclaiming it when conditions improve. If evolution on Earth produced organisms that can toggle water dependence on and off, what might evolution produce elsewhere? On worlds where water never existed, where the only liquid has always been methane or ammonia or something we have not imagined, life adapts to what is available. It exploits whatever energy gradients exist. It builds with whatever materials the environment provides. Water happens to be abundant here. But abundance does not equal necessity. The philosophical implications cut deeper than the practical ones. For centuries, we have defined life by what we know. Cells, DNA, proteins, metabolism, reproduction. These definitions work for Earth. They describe every organism we have encountered. But they are descriptive, not prescriptive. They tell us what life is here, not what it must be everywhere. If we find something on Titan that maintains chemical disequilibrium, that harvests energy, that reproduces information, but does it with entirely different molecules in an entirely different solvent, do we call it life? Or do we invent a new category? The answer matters, because it determines whether we are alone or surrounded, whether biology is rare or common, whether Earth is special or typical. The search continues, not just in space, but in laboratories. Researchers are synthesizing alternative biochemistries from scratch, building artificial cells with non-biological membranes, testing whether RNA can function in ammonia, whether silicon-based polymers can store information, whether metabolism can operate in supercritical carbon dioxide. These experiments are not trying to create life. They are mapping the boundaries of what chemistry allows, defining the limits of the possible. And so far, those limits keep expanding. Every assumption we test reveals flexibility we did not expect. Every constraint we thought was absolute turns out to be negotiable. Water is important for Earth life, critical even. But the universe is not obligated to repeat our solution. It has had billions of years to experiment, trillions of worlds to work with, countless combinations of chemistry and energy and environment. Some of those experiments probably failed. Many may have produced nothing. But if even a fraction succeeded, using pathways we have not considered, in conditions we have not explored, then life is more common and more strange than we ever imagined. The detection problem looms larger than most people realize. If methane-based life exists on Titan, our current instruments might pass right over it. We search for biosignatures based on Earth chemistry, oxygen in atmospheres, methane ratios that suggest biological production, phosphorus in specific compounds. All of these assume water-based metabolism. A methane organism would leave completely different traces. It might consume hydrogen instead of oxygen, produce acetylene instead of carbon dioxide, build membranes from molecules we have never classified as biological. Our telescopes and probes are tuned to detect what we already know, not what we have yet to imagine. This creates a detection paradox. The more alien life becomes, the harder it is to recognize. We could be staring directly at biosignatures without realizing they are biosignatures at all. Scientists are now working backward from this problem. Instead of asking what life should look like, they are asking what chemical disequilibrium looks like. Any living system, regardless of its chemistry, must maintain itself away from thermodynamic equilibrium. It must concentrate certain molecules while depleting others. It must create order in a universe that favors disorder. These principles apply whether the solvent is water, ammonia, or liquid nitrogen. Thermodynamics does not care about the details. It only cares about the energy flows. So researchers are developing detection methods based on disequilibrium itself, looking for environments where chemistry is too organized to be random, where concentrations are too precise to be accidental, where patterns persist despite conditions that should erase them. This approach is agnostic. It does not assume carbon or water or anything specific. It simply looks for the fingerprint of life, the maintenance of improbable states through energy expenditure. The next generation of space missions carries these principles forward. Dragonfly, scheduled to explore Titan in the 2030s, will not just search for water-based life. It will analyze organic chemistry in methane lakes, test whether complex molecules are being synthesized and organized in ways that suggest metabolism, look for spatial patterns in chemical distribution that random processes cannot explain. Europa Clipper, launching soon, will probe beneath the ice of Jupiter's moon, not just searching for water, though water is there. Searching for energy sources, chemical gradients, anything that could power metabolism, water-based or otherwise. The mission design reflects a broader philosophy. Do not assume. Measure everything. Let the data reveal what is actually happening rather than confirming what we expect to find. This shift matters beyond astrobiology. It changes how we think about life itself. For centuries, biology has been a historical science. We study what evolution produced here. We categorize it. We trace lineages. But we cannot experiment with different starting conditions. We cannot rerun evolution on other worlds to see what else is possible. Alternative biochemistry offers a way around this limitation. By exploring what chemistry allows, not just what evolution chose, we map the space of possible biologies. Some regions of this space are easy to reach. Carbon and water sit at the center, accessible under common conditions. Other regions require exotic environments, high pressure, extreme cold, unusual solvents. But the space itself is vast and mostly unexplored. Early results surprise even the researchers. Amino acids, the building blocks of proteins, remain stable in concentrated sulfuric acid for weeks. Not all of them, but most. The molecular backbone survives even when side chains degrade. This was unexpected. Sulfuric acid destroys most organic molecules instantly. Yet these fundamental biological components endure. Venus suddenly becomes more interesting. Its clouds contain sulfuric acid at concentrations that match the lab experiments. Temperatures there reach 337 degrees Celsius. Hot enough to sterilize by Earth standards. But if amino acids can survive in that acid, what else might persist? Could metabolism operate at those temperatures using acid-resistant chemistry? Could information storage work with different molecular scaffolds? The questions multiply faster than the answers. Each experiment opens new possibilities. Each possibility demands new experiments. The cycle accelerates. Meanwhile, synthetic biology pushes from another direction. Researchers are not just theorizing about alternative life. They are building it. Or trying to. Artificial cells with membranes made from non-biological molecules. Genetic systems based on synthetic nucleotides that do not exist in nature. Metabolic pathways using enzymes that evolution never produced. These are not natural organisms. They are proofs of concept. Demonstrations that the principles of life, information storage, metabolism, reproduction, can be implemented in multiple ways. The chemistry is negotiable. One team created a synthetic cell membrane that functions in methane at -180 degrees Celsius. Not a living cell. Just the outer boundary. But functional. It maintains separation between inside and outside. It allows selective transport of molecules. It can grow and divide when energy is supplied. All the basic requirements of a cell membrane, achieved without phospholipids or water. Another group developed an RNA-like polymer that remains stable in ammonia. It can store genetic information. It can fold into shapes that catalyze reactions. It can replicate when provided with the right building blocks and energy. It is not RNA. The backbone is different. The bases are different. But it performs the same functions under completely different conditions. These synthetic systems are crude. They do not compete with natural biology in complexity or efficiency. But they prove the concept. Life's core functions can be separated from life-specific chemistry. Information, metabolism, reproduction, these are abstract processes that can be instantiated in many materials. Water-based carbon life is one solution. Not the only solution. The implications extend beyond planets and moons. Interstellar space contains complex organic molecules. Clouds of gas and dust where chemistry happens slowly over millions of years. Temperatures there approach absolute zero. No liquid water. No liquid anything by conventional standards. Yet molecules form. Amino acids. Sugars. Nucleotide precursors. Complex polymers. Some researchers speculate that prebiotic chemistry might operate even there. Not life, but the chemical steps that precede life. Building blocks accumulating, organizing, waiting for conditions that allow the next stage. If chemistry can progress that far in frozen space, what happens when those molecules encounter environments with more energy? When comets deliver them to planets? When they settle into subsurface oceans or methane lakes? Do they simply dissolve and disperse? Or do they find pathways toward organization? Toward metabolism? Toward something we might eventually call life? Nobody knows. But the universe has been running this experiment for billions of years across billions of star systems. The sample size is astronomical. Even low-probability outcomes become near certainties given enough trials. Which brings us back to the original question. Could life exist without water? The answer is no longer speculative. It is mathematical. Chemistry allows it. Thermodynamics permits it. Evolution, given time and opportunity, explores every available niche. Water is common, but not universal. Many worlds lack it entirely. If life is restricted to water, then life is rare. Most of a universe is off-limits. But if life can adapt to other solvents, other chemistries, other conditions, then life is potentially everywhere. Every liquid environment becomes a possibility. Every energy gradient becomes an opportunity. The universe transforms from mostly dead to mostly alive. We just have not learned to recognize it yet. That recognition might come soon. Or it might take centuries. But the search has fundamentally changed. We are no longer looking for Earth 2.0. We are looking for any world where chemistry becomes biology. Where molecules organize. Where energy flows through systems that maintain themselves against entropy. Where information persists and replicates. The definition is broader now. The criteria more flexible. And the target list grows longer every year. Titan. Europa. Enceladus. Venus. Mars. Worlds in other star systems with thick atmospheres and exotic chemistry. Even objects we have not imagined yet. Each one represents a test. A natural experiment in what life can be. Some tests will fail. Most probably. But the universe is patient. It has time. And it has more worlds than we can count. Somewhere in that vast laboratory, chemistry is doing something remarkable. Something we have never seen. Something that redefines what living means. We just have to keep looking. Keep testing. Keep expanding our assumptions about what is possible. Because life, it turns out, is more stubborn than we thought. More creative. More willing to exploit whatever the universe provides. Water is not the universal solvent for life. It is the convenient solvent. The one we know. The one we understand. But convenience is not necessity. And understanding is not limitation. The cosmos is full of liquids we have barely studied. Full of chemistry we have not explored. Full of possibilities we have not considered. Life exists without water somewhere. Maybe many somewheres. We are only beginning to learn how to find it. The search for life beyond water is not just about finding organisms on other worlds. It is about rewriting the rules we thought governed existence itself. Every assumption we started with, carbon is essential, water is irreplaceable, DNA is the only genetic code, has revealed cracks under scrutiny. Not failures. Just limitations in our imagination. When we look at Earth, we see one solution to the problem of being alive. A solution refined over 4 billion years. Optimized for this planet's specific conditions. But optimization for one environment does not mean universal superiority. A polar bear is perfectly adapted for Arctic survival. Drop it in the Sahara and it dies within hours. Earth life is the polar bear. Magnificent in its domain. Potentially helpless elsewhere. The question is not whether life exists without water. The question is whether we are capable of recognizing it when we find it. Our instruments measure what we tell them to measure. Our experiments test what we design them to test. Our theories predict what our assumptions allow. If those assumptions are too narrow, we create blind spots. Not in space. In our own thinking. Consider what we have learned just by looking carefully at Earth. Tardigrades that survive complete dehydration. Bacteria that thrive in boiling acid. Archaea that metabolize hydrogen sulfide in total darkness. Life that should not exist according to the rules we wrote before we found it. Each discovery forced an expansion of what we thought was possible. Each expansion revealed that life is more flexible, more resourceful, more stubborn than we imagined. Now multiply that by every world in the universe. Every combination of chemistry and energy and time. Every liquid surface, every subsurface ocean, every atmospheric layer where molecules can meet and react. The sample size is not hundreds or thousands. It is trillions. And we have examined perhaps a dozen in any meaningful detail. Titan is not just a moon. It is a laboratory where chemistry operates under conditions that would destroy everything familiar. Yet chemistry continues. Molecules form. Reactions proceed. Organization emerges. Whether that organization has crossed into biology remains unknown. But the fact that it approaches the boundary tells us the boundary is not where we thought it was. Venus is not just a hellscape. It is a test of whether life can adapt to environments that seem fundamentally hostile. Sulfuric acid clouds should be sterile. Yet amino acids survive there. Not for seconds. For weeks. The chemistry does not care what we think should happen. It only cares what thermodynamics allows. And thermodynamics is far more permissive than our intuitions suggest. Europa and Enceladus are not just frozen moons. They are active worlds with liquid oceans, chemical energy, and organic molecules. Everything life needs, assuming life is what we think it is. But even if it is not, even if those oceans harbor something completely alien, the conditions exist for chemistry to become complex. For complexity to become organized. For organization to persist and adapt. Mars is not just a dead planet. It is a world in transition. Once warm and wet. Now cold and dry. Whatever lived there, if anything lived there, faced a choice. Adapt or die. If it adapted, it did so by changing its internal chemistry. By using different solvents. By finding energy in new ways. By becoming something we would not immediately recognize as alive. These are not distant abstractions. They are places we can reach. Missions are planned. Instruments are being built. Samples will be collected. And within our lifetimes, we will have data from worlds that might harbor non-water life. The data will not come with labels. It will not announce itself. It will be ambiguous. Puzzling. Open to interpretation. And that is when the real work begins. Proving that something is alive when it uses completely different chemistry is not straightforward. It requires ruling out abiotic explanations. Demonstrating complexity that random processes cannot produce. Finding patterns that persist despite thermodynamic pressure to dissipate. This is harder than it sounds. Nature produces organized structures without life all the time. Crystals. Convection cells. Chemical oscillations. The line between complex chemistry and simple biology is not sharp. It is blurred. And we are still learning where to draw it. But the effort matters. Because the answer determines our place in the universe. If life requires water, we are probably rare. Water is common, but the right amount of water in the right place at the right temperature for the right duration is not. Earth might be an outlier. A statistical anomaly. We might be alone, or nearly alone, in a vast sterile cosmos. If life can use other solvents, the equation changes completely. Suddenly most worlds become candidates. Not just planets in the habitable zone, but moons around gas giants, rogue planets in deep space, even objects in interstellar clouds where chemistry proceeds at glacial pace but time is unlimited. Life becomes probable instead of miraculous, common instead of rare. And we transform from lonely observers into one voice among many. That transformation is already underway. Not through discovery yet. Through possibility. Every time we expand the conditions life might tolerate, we expand the number of places it could exist. Every time we demonstrate alternative chemistry in the lab, we prove that nature has options we did not account for. Every time we find something on Earth surviving in conditions we thought were impossible, we realize our map of the possible was incomplete. The search is not waiting for better telescopes or faster rockets. It is happening now in laboratories where researchers build synthetic cells from non-biological materials, where they test whether information can be stored in molecules that evolution never used, where they explore whether metabolism can operate in solvents that would kill Earth life instantly. These experiments are not trying to create aliens. They are mapping the landscape of what chemistry allows. And that landscape is far larger than the small region Earth life occupies. This is what makes the question urgent. Not because we will find an answer tomorrow, but because every year we delay is a year we spend looking in the wrong places with the wrong tools, asking the wrong questions. The universe is not hiding life from us. We are hiding it from ourselves by defining it too narrowly. By insisting it must look like us, behave like us, use the same molecules we use. Life does not care about our definitions. It cares about energy gradients and chemical stability and reproduction rates. It exploits whatever works. Water works here. Something else works there. The chemistry is negotiable. The principles are not. When we finally find life that does not need water, it will not feel like a triumph. Not immediately. It will feel like confusion. Like data that does not make sense. Like results that contradict everything we expected. And we will spend years arguing about whether it is really life or just complex chemistry that mimics life. The debate will be fierce. The evidence will be ambiguous. And the answer will reshape how we understand existence itself. But that moment is coming. Not because we are clever. Because the universe is vast. And in that vastness, chemistry has had billions of years to explore possibilities we are only now beginning to imagine. Some of those explorations led nowhere. Dead ends in a space of possible biologies. But others, perhaps many others, found pathways we never considered. Solutions we never predicted. Forms of life that would astonish us precisely because they are so unlike anything we know. We are not alone in the universe. That seems increasingly certain. But we might be alone in our particular solution to the problem of being alive. Alone in using water and carbon and DNA. Alone in our specific implementation of the abstract principles that define living systems. And that makes us both ordinary and unique. Ordinary because we are one example among many. Unique because every example is different. The cosmos does not repeat itself. It explores variations. It tests alternatives. It tries every combination until something works. And when something works, it persists. It adapts. It spreads as far as conditions allow. Water-based life spread across Earth. Methane-based life might spread across Titan. Sulfuric acid life might float in Venus's clouds. Each one thinks it is normal. Each one assumes its chemistry is the chemistry. None of them realize they are variations on a deeper theme. We have the chance to see that theme, to recognize the pattern beneath the diversity, to understand what life is when you strip away the accidents of Earth's particular history. That understanding will not come from philosophy or mathematics. It will come from observation, from data collected on other worlds, from chemistry tested in conditions we thought were impossible, from organisms, if they exist, that challenge every assumption we carry. The search continues because the question demands an answer, not for practical reasons, not because it will change daily life or solve immediate problems, but because it redefines what we are and where we fit in the cosmos. And that redefinition is already happening. Right now. In laboratories and on spacecraft and in computer models that simulate alien biochemistries. Water is not the universal solvent for life. It is the local solvent. The one we know intimately because we are made of it. But the universe is not local. It is incomprehensibly vast. And in that vastness, other solvents flow. Other chemistries operate. Other solutions to the problem of being alive persist in conditions we would find lethal. They are out there, waiting to be found. Or perhaps they are already here, in data we collected but did not understand. In samples we analyzed but misinterpreted. In signals we detected but dismissed. Because we were looking for ourselves. And they are not us. They never will be. That is exactly what makes them worth finding.